thank you very much, everybody, for coming on this dark winter's night. I have entitled my talk Viking Genes, and what we're going to talk about is a series of highlights from some of the work that I've been doing over the last 15 years in Orkney and in Shetland. These stem from the Orkney study in Orkney and the Viking study in Shetland that many of you may have participated in, and I'm going to end by discussing the Viking 2 study that I'm here to launch. So we're going to talk about cohort studies. What is a cohort study? Why am I studying the Northern Isles? And then we're going to go into some of the things we can learn from these kinds of studies. First, looking back in time, what can we learn about the past from our DNA? What's so special about the gene pool here in Shetland? And then I'm going to start looking forward into our own futures to see what we can learn about health and disease with a few examples taken from our studies. So let's start with cohorts. Many of you will know that the word cohort means a group of people who share something in common. But it's actually over time evolved a new meaning such that the thing that they share is that they are part of a study, part of a cohort study, which is essentially a rich collection of samples and data, like the Orkney study or the Viking study. And you get different kinds of cohort studies. Case control studies are famous. You collect 1,000 or 10,000 examples of people who've had a heart attack or perhaps breast cancer, and then 1,000 or 10,000 controls, people who are similar to the others but do not have the disease in question. Better, in my opinion, are population-based studies where you recruit a random sample of the population, as we did in Viking. This allows you to study any health outcome that occurs in your population. You get much more for the money that your funding body put in. You also get family-based studies and a variety of different studies. The most famous cohort study and the original cohort study was the Framingham Heart Study here that many of you may have heard of. This was founded in 1948 in Massachusetts in the USA, and it's responsible for many of the things that we take for granted now, knowing, for instance, that smoking increases your risk of heart disease, and such things were discovered in the Framingham Heart Study, which is a cohort study. So what do we do in these cohort studies? We first collect samples. So you might collect whole blood, you might derive blood plasma from that, DNA, urine, cells, all sorts of biological tissues that you might want to make measurements on in the future. You also need to acquire some data, typically with questionnaires asking people about their health and behaviors, but also possibly physical measures, behavioral measures, blood biochemistry from the blood sample. You can annotate these samples with so-called omics technologies, where you measure hundreds and thousands of metabolites or hundreds and thousands of proteins or other molecules in the blood or in any other tissue. And finally, many people now can link their cohorts to electronic health record data. So these are data that exist in the National Health Service when you interact with the National Health Service, and it enriches cohort studies to be able to link to that. Of course, the cohorts that I study also have a large genetic component. So this involves reading the DNA, either quickly running through it using a gene chip technology, all the way through to reading every single letter in your genomes, sequencing your whole genome. Obviously, these complex studies involving health data from living subjects have a lot of governance and ethics attached to them. There are complex rules and lots of scrutiny that we have to go through. It's almost a full-time job to keep on top of all our ethical approvals. These days, people are no longer happy with the word cohort studies, and as they get bigger and bigger, particularly in the case of the UK Biobank, a study of 500,000 people, principally in England, they start using the word biobank for the very large mega cohorts. So that's what a cohort study is. I just want to remind you, or those who don't know, about the cohort studies that we ran. I started all this work in March 2004 in Orkney, in the Orkney Complex Disease Study, and we recruited over 2,000 people there. They were volunteers, so their mean age, average age, was about 50, in their early 50s, at the point of recruitment, and there was a 60 to 40 ratio of women to men, because women are just better at coming forward and volunteering for these things. So having learned how to run these studies, we then raised more money and came and ran the Viking Health Study here in Shetland. We recruited 2,000 people again, this time over the course of 2 years, because we'd streamlined what we did. It won't surprise anyone in this room to be told the history of Orkney and Shetland, in particular that the Norsemen and women to some degree crossed the North Sea and colonized these places, the Northern Isles, the Western Isles, following the sea road down to Man and eventually Dublin. So these populations are part Norse and part Scottish. And indeed, when we analyze their DNA, we can see this. This plot that you see here on the right is a plot of a genetic space. So each dot is a human being who has had a genetic test done, and how close together these dots are tells you how closely related the individuals are. And you will see that people from a given population tend to cluster together. Here we have Spanish, here we have Tuscans from Italy, so you've got a sort of Mediterranean cluster here. Over here we have Finns at a different side, and here in the middle are the British. But you can see that Orkney and Shetland are distinct from all other European populations, including the British population, but that Shetland is much more distinct, even on a European scale. Yet, Orkney and Shetland are clustered together and are each other's nearest genetic relatives. Another thing that we have a lot of in the Northern Isles is families. So in our studies, we had many nuclear families, parents, offspring, siblings, but also people who are much more distantly related to one another. This is a real pedigree from one of my studies. So the people in black here have joined the study. Here you have a pair of sisters. Their father, their mother, and their granny have joined the study. Here are other people who have joined, but they're all linked, perhaps unbeknown to them, through a large, deep, and complex pedigree. This is very useful for certain aspects of genetics. So you're studying inheritance. It's always better to have a family so you can see the genes coming down the family. It's particularly useful for studying rare variants, which are variants that not very many people in the room will carry. And I'm going to talk quite a lot about these in the course of the lecture this evening. So what did we measure? We mostly measured everything that you could measure about someone without making them squeal. So lots of measures that you may have had from your doctor: blood pressure, cholesterol, blood sugar, rather commonly used measures, but also much more rarely used measures, research measures, such as photographs of the back of the eye, whole body scans in Orkney so that we could look at body composition. I learned that I have a kilogram of fat in my head, which was news to me, but apparently is quite ordinary. So there are all sorts of amazing things that we measured, covering a very broad range of risk factors for disease and other things that are not known to be risk factors, but nevertheless may be genetic and may therefore be interesting. I'm just going to take you through a couple of these. This is a picture of a retina, so the layer of cells that are light sensitive in the back of your eye. And we're able to take a photograph of this by taking the photo through the lens of your eye. And the reason we do this is to look at these vessels that are microvessels, small arteries and veins that are providing blood and oxygen to your eye. This is actually my retina here, and you can see that some of my vessels are wiggly and some of them are straight. So you can measure things about these vessels. The eye is the only place in the human body where you can directly see these vessels non-invasively. And these vessels grow embryologically from some of the same places that the vessels in the brain grow. So they may be very informative with regard to stroke and other such diseases. So we take these images and we can put them through a computer analysis program to zoom in on them, and you can use the pixels to measure the widths, so the caliber of the vessels, the angles that they branch at, and all sorts of other things that you probably don't ordinarily think about in terms of your health. And some of these have already been related to differences in blood pressure, but we're interested to learn more about them, and I'll tell you more about that later. Another thing that's very important to these studies is not to just plan it for today, but to plan it also for tomorrow, to try to future-proof your study by collecting all that you can. Lots of aliquots of blood, lots of different tissues, urine, plasma, and so on. We have freezers and freezers, a room full of freezers in Edinburgh, full of all these tubes of plasma. So this is so that when next year someone discovers some amazing molecule that's absolutely critical in diabetes or in cancer or whatever else, we have the material in the freezer. We don't have to come back to get everybody, and we can start measuring this, trying to understand the distribution, seeing if there are genetic contributions. It means that when technology develops, as it does every year, we have the material waiting to be used. Another thing that we can do in Scotland, much better than anywhere else in the world thus far, is to link the data from our cohorts to the data that the National Health Service has. And this is called electronic health record linkage. So we have everyone in our studies who consented to this is linked to their health data. So we know, for instance, when they have a hospital admission, why they were admitted, what happened to them. We know about the maternities, we know about cancer registrations, we know if they encash a prescription at a chemist, and we're going to get linkage soon to blood biochemistry, so that if you're, for instance, a diabetic, you're going to get your hemoglobin A1C measured to check your glycemic control. We get all these measurements. It enriches the cohort data in an amazing way because it goes backwards in time to about 1985 and it goes forwards in time all the way. So we essentially, without coming to see everyone again, we find out what happens to them as they get ill and eventually one day in the future die. It's a very powerful approach to make use of our National Health Service, to take these data and put them to work so that we can understand biology and disease better. So people often ask me why I study the Northern Isles. It's not just as an excuse to come up to Shetland and Orkney for a visit, much as I like doing that. Why don't we study the good burghers of Edinburgh here where we work? Well, there are a variety of reasons, mostly to do with the fact that these populations are supposed to be isolated populations. That is, they descend from a small number of founders. The number of people who started the population of Shetland is much lower than the number of people who started the population of Glasgow. And also fewer people have migrated here in the meantime. Of course, people have come, people have gone, but the number is much lower than in other places. Marriage, therefore, tends to occur within the community. And this creates a certain number of factors that are very interesting from a geneticist's point of view. Essentially, I think it can be summarized by saying that the population should develop its own gene pool that's subtly different from the gene pool of neighboring cosmopolitan populations. What actually happens is that many genetic variants are lost. If none of the founders of the Shetland population carried a particular variant, and no one who emigrated here since carried it, it's not going to be present here. So there's much variation that's found elsewhere that is not present here. That's less interesting than the other side of this coin, which is that some of the variants that are here ought to have increased in frequency. So they may be very rare elsewhere, but they may be rather common here, and that could be important for our health. This is called genetic drift. The frequencies of these variants drift upwards or downwards through time. It's a drifting process, generation to generation. And that's why we're interested in isolated populations. And it's no wonder there's genetic drift, actually, when we look back into the marriage records. I must apologize, this is for Orkney, but this work has not yet been done for Shetland. You may recognize the map of Orkney here, with the North Isles here, the mainland of Orkney here, and the South Isles here. And what has been done is that for every marriage in 1861, people have counted the proportion of the people who are contracting marriage that were born in the same parish. So it varies. It wouldn't surprise you that the most isolated and outlying island, North Ronaldsay, about 90% of marriages were contracted between people who were born on that island. And I can contrast this with Kirkwall, the capital of Orkney here, where only 10% of marriages were contracted between people who were born in Kirkwall. But you can see that there's a general trend that the Isles, so Westray, Sandy, Rousey, Hoy, South Ronaldsay, to some degree Stronsay, are more endogamous. They have more within community marriage, but also the more outlying parts of the mainland, Bursey here in the West Mainland, Harry to some degree, and Derness here, whereas there's a sort of axis here between Kirkwall and Stromness, including Finstone, where there's much more movement going on. People are moving around a bit more and not marrying so much within the parish. So marriage was very local, even 120 years ago. It's not that long ago. Of course, this is breaking down as we move into the modern age of mass movement, but still some of these patterns persist in our DNA. And you probably won't have heard of Eric Lander, a very famous geneticist who was involved with the sequencing of the human genome. He became interested also in isolated or drifted populations, saying that they should thus make it easier to discover some disease-associated genes. And indeed, he said we should hit the jackpot with isolated populations. And I'm pleased to report that I feel we have. In the last 15 years, we have published more than 220 peer-reviewed articles on either the Orkney or the Shetland population or both. Many of them in the top journals, Nature Genetics. But today, I'm going to give you a few highlights of papers which were published in 2019 only. So this is the most recent research that we've done. And it varies from rare diseases through to common variants, through to history, and something about the uniqueness of Shetland. So a very broad range of research questions can be answered by the sort of very rich data sets that we are collecting. So let's first look backwards. Let's think about our genetic ancestry. What I'm particularly interested in is what's known as population genetic structure. So how does the population break up into clusters? If I took you in the room, who's genetically a cluster, who's another cluster? How are these clusters related to one another? Where are they found? What's the pattern of genes across the landscape? What's the pattern of genes across geography? Is it all blended together, or can we see differences as we move from place to place? And beyond that, why is that the case? Are there different contributions to each population from different source populations, or is it all the same? This part of the talk is based on genome-wide data, so we're looking across all of the different ancestors. To do this, first we put together a large sample, 2,500 people from a variety of different studies all across the British Isles and Ireland. Scotland, England, Ireland, Wales, various islands, the Hebrides, the Isle of Man, Orkney and Shetland. But these people were not a random sample. They were a very specific subgroup because all of them had 4 grandparents from one county. So born typically within 50 miles or so of one another. But in Shetland and in Orkney, we cranked this up a little bit further because I demanded that all the volunteers had 4 grandparents from one isle or one parish, so that we could look even more fine scale to see if we saw patterns in the data. So this is actually a small subset of people, perhaps 10% of all the people that we have. We have something like 30,000 samples, so a much smaller proportion fitted these criteria. What did we find? Well, we put this through a rather complex analytical program in the computer, and it determines genetic clusters, and it actually found 42 clusters in the data, as you can see here. And the first thing I want you to notice is that the biggest differentiation here in this tree, which is showing how people are related. If they're close together, they're closely related. If they're far apart, they're much more distantly related. And the biggest difference in the whole of the British Isles and Ireland was Orkney and Shetland versus everybody else. So there really is a signal here of isolation, of genetic differentiation. If we look and label the other parts of the tree, we see a branch over here with England and a branch with Wales that are related to one another. Then we have Ireland here and Scotland here and the Hebrides here that all form a branch with each other, and that deeper links in to England and Wales. Then if we break down Scotland a little bit, we can see there's a northern and eastern cluster. For instance, Aberdeenshire, Tayside, Inverness, and so on. And then there's a southern and western cluster. And within the southern and western cluster, we find the Ulster people, the Ulster Scots, and also the Manx. So they are genetically part of the Scottish cluster. Another way of looking at this, rather than just looking at the tree of relationship, is to plot these on a map. And I hope you can make this out, but each dot is an individual human, and they are plotted at the sort of average position of the birthplace of the 4 grandparents. So if your one grandparent's born here, your other one there, So here, 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 and here, you get plotted there in the middle. And it's amazing, you see this very large English group in dark blue, showing that there's no further structure in England. They have been moving around within this region, but the whole rest of the country is structured into lots of different subgroups. And it's amazing how geographically circumscribed these places are. We rediscovered the structure that's present in England and in Ireland that had already been shown, but we discovered for the first time that the Isle of Man was its own genetic cluster, and therefore definitely worthy, in my opinion, of further study. We discovered this broad south-west to north-east divide in Scotland that had never been seen before. For part of it, it tends to follow the River Forth, but then I don't have quite enough samples to see where this ancient boundary appears to fall. We also saw, as I said, these links between southwest Scotland and Northern Ireland, northeast Ireland at least, probably due to the plantation of Ulster. And finally, we saw that the Hebrides were also an isolated population, as you might expect, but it's a different thing to expect it than to show it genetically. And remember, these divisions are visible using DNA alone. I did not tell the computer where these guys came from. It found the clusters. I then plotted them on the map. So it's discovering these patterns from the patterns in our DNA. But let's look now at the more interesting part of the world, the Northern Isles. And it's actually incredible to me, the degree of fine-scale structure, the degree of differentiation that you see here, because different islands can be told apart using DNA alone. Let's look at Orkney first. We see clusters in Westray, North Ronaldsay, Sandy, Stronsay, South Ronaldsay. There's an East Mainland cluster, a Hoy cluster, a West Mainland cluster, and the parish of Bursey separates even from its neighbours in the West Mainland. Rousey is also a cluster, Rousey linking in with the West Mainland. So the Ainhallow Sund is a channel here between Evie and Rousey. It's a mile wide, and we can tell people where they come from using DNA alone over just a mile. And indeed, we can do exactly the same thing in Shetland at Clift Sund. I measured Clift Sund on the map. It's less than a mile wide. So we can tell people from Wester Quarf apart from people from East Borough just using DNA. So there are very, very fine scale patterns to be found. It's very similar here. We can separate folk for Unst and Yell, folk for Walsa. There's a North Mainland cluster, a West Side cluster. We have a cluster relating to Central Shetland, so boot hands with Tingwall and so on. We have a cluster that relates Coningsborough and Sandwick, a Dunrossness cluster, which Fair Isle is a side group of, and finally here we have the Borough cluster. So it's incredible how yet, despite the movement that has happened, we can still pick these things out in DNA. But we won't be able to for very much longer. We actually had to use more of the older people in our studies to pick this up, because the younger generation, there are far fewer people that have all four grandparents from one place as people begin to move around and they start to dilute these interesting patterns. Now, with such a rich data set, there are many different ways to look at it. Sometimes you don't want to impose a tree structure on things. It's not really true that populations totally split and never ever meet again. So we can rather use this principal components approach. Again, each dot is an individual, and how close together they are tells you who's related to whom. So we pick out the same patterns again with Ireland, Scotland, England, and Wales. And here we have Orkney and Shetland sticking out away to the side, very different from everybody else. It's interesting, actually, there's a few dots here that are people from around about Tingwall who are showing a slight hint of what looks to be English ancestry. Whether that's Dutch, because we don't have Dutch in here, truly English, or some part of Scotland that looks like England, I do not know. But it's intriguing and had never been seen before. But what I want you to focus on is actually rather these green diamonds that are here. These are people from Caithness and Sutherland. And the fact that they're pulled out to the left of this plot, away from the rest of the Scots, shows that they too have Orcadian or Shetlandic ancestry. And of course, many of you will know that Caithness and Sutherland used to belong to the Earldom of Orkney. And probably, definitely, there's a lot of exchange between South Ronaldsay. These other red things here are South Ronaldsay and Orkney, the nearest island to Caithness. But we're seeing here genetic evidence of input from the Northern Isles into the mainland. Also up here, with the red and blue X's are the Hebrides. And what this is telling us about them is that they also have a little bit of Northern Isles ancestry, probably going back to at least the Viking Age or perhaps before, but also they've been pulled here away from the rest of Scotland towards Ireland. So they also have an Irish component in their ancestry in the Hebrides. So that's the structure as we see it. What about how this structure came to pass? It always amuses people when I give talks to hear of what the Orcadian, or the men of Kirkwall at least, get up to when everybody else is preparing or eating their Christmas lunch. And it's not that much more unusual than what the men of Shetland get up to at many different uphelias around about this time of the year. So is this because of Norse heritage? I think we know the answer is yes. But we had richer data than ever before, so we used a different algorithm this time that models the genomes of the Shetlanders and the Orcadians and the Irish and so on here as being made up of the genomes of people from continental Europe to try to get a picture on where the ancestry might have come from. So we had continental Europeans from Spain, Italy, France, Belgium, Germany, Poland, Denmark, Sweden, Norway, and Finland to sort of try to cover all the major possible source populations that are near to the British Isles. And you can see here that across all these populations, there's a very large red component, which is a French-like component. And I'm not for a minute suggesting that we're French, but it may be a proxy of some more ancient Celtic ancestry. This is probably the oldest component that we're seeing here. Then also shared across all populations, but noticeably greater in England, over here on the left, is a German-like component, probably brought over by Anglo-Saxons, but probably also brought by others, both before and afterwards. And then finally, I'd like to highlight this dark blue component here that's noticeably larger in Ireland and in Orkney and in Shetland. That is a Norwegian-like component. Note also here a little bit of Danish among the English. So this is a first look. We decided to look at this in a little bit more detail. So here we have Orkney and Shetland, again divided up into these genetic clusters that we've discovered. And we're allowing this time for there to be English, Welsh, and Scottish sources for these populations, as well as some continental sources, to try to get at this age-old question of the Scottish to Norse sort of percentages in this population. You'll be happy to hear, I suppose, that Shetland beats Orkney. Just. So it's coming out at, I don't know, call it 25% or so, and then Orkney's a little bit lower. But you'll see there are actually significant differences between different parts of Orkney and Shetland. Yell and the west mainland of Shetland are the most Scandinavian of any place outside Scandinavia, reaching 28% Scandinavian. And in Orkney, it's North Ronaldsay and Bersey that are the highest. In Orkney, the lowest, perhaps not surprisingly, is South Ronaldsay. There's a lot of movement between South Ronaldsay and Caithness, which will have diluted out the signal. In Shetland, the lowest is Fair Isle, and also this sample from central Shetland. I excluded everyone from Lerwick from this analysis because I don't believe it actually exists as a population. It underbreeds and people move in all the time. So these are people from a Tingwall and so on. But even in Fair Isle and in Tingwall and in South Ronaldsay, they have much more Norse component than the next highest, which is the Hebrides and the Isle of Man. Again, I note there's this quite large English component showing up in the Tingwall people. So there's definitely something, but there's English components here as well that may reflect a deep British, pre-Anglo-Saxon ancestry, or who knows what. It's something that needs to be looked into. We decided to try and zoom the analysis in even further, and instead of just saying Scottish or Norwegian, to ask where in Scotland, where in Norway. So we did that again using our genetic clusters that we've discovered here for Orkney and Shetland. So taking the Scottish side first, we have a very large component coming from Perthshire, Angus, and Fife, between 30 or 25 and 50% in every subpopulation. And this might not surprise some genealogists amongst you. There are a few families who have genealogies going very far back. So the Spence of Scapa family in Orkney is a very old family, mostly in Bursey these days, and we know that that ancestor came from this part of the world. My own family, the Wilsons of Fair Isle, also came from Fife, probably in the 1580s to Scalawa. So we have a few examples where we have the genealogy going far enough back, and I would predict that many of the Scottish settlers came from what's now Tayside and Fife. We also see input from Aberdeenshire, from Inverness Shire, and a few other places that were too small to fit onto the graph, including a little bit of input that this models as coming from England. But it may come from somewhere like England, such as Holland, because we did not include Holland in this analysis. So what about the Norse side of things? Most of the Norse component is from a group called Nor 10, and there's a little bit coming from Nor 9, and there are smaller bits coming from other places. So where do these come from, you might ask? I'm very happy to report that there's also genetic structure in Norway, just as there is in Britain, because if all of these pie charts look the same, we wouldn't be able to tell where in Norway our ancestors came from. But you can see here the Swedes have their cluster. Here in Zealand is a different cluster. Germany's different down here. And all across Norway we have different clusters. This sort of light blue one up here in Finnmark and Nordland, all the way down the west coast. And Nor 9 is actually this bright purple cluster here that's only really seen in Hordaland and so the county around Bergen, and also in the county to the north called Sonafjordene. And Nor 10 is found here pretty much only in Hordaland. So it looks like the ancestors, the Norse ancestors of the Shetlanders, did not particularly come from all over Norway, but were very much focused in this region. The third most common component is this Nor 13 group here in bright lilac that comes from Rogaland, near Stavanger. So we've talked about the overall picture, looking across all our genes. I now want to focus in on some lineages. So this is a real family tree of a collaborator of mine, and you see that eventually it becomes a complete morass. It's so complex, and everyone is related to everyone. But we can use two special bits of DNA to try to disentangle this web. That's one called the Y chromosome. It's just a block of DNA that's inherited from father to son to grandson down the generations. Only men carry this, and it's inherited a bit like a family coat of arms. It's the same coat of arms, but it changes a little bit each generation. And you can use this to build family trees. Not family trees using paper, but family trees using genes. There's another piece of DNA called mitochondrial DNA, or mtDNA for short, which is very similar. It's a block of DNA, but it's inherited down the mother line, not the father line, from mother to her children. I inherited my mother's mtDNA, but I did not pass it on to my children. They have my wife's mtDNA. So you look at these two lineages and you can disentangle to some degree what's going on. Just to emphasize it a little bit more, here is my pedigree in the shape of an Ahnentafel here. This is me. Here's my Y chromosome. Here's my dad and mum, my grandparents, my great-grandparents, and I got my Y chromosome from dad, who got it from granddad, who got it from my great-granddad, Jerry Wilson, from Springfield in Fair Isle. Here's my mitochondrial DNA, which I got from my mother, from her mother, from her mother, who is Bina Tullach, whose family came from North Ronaldsay in Orkney. So you're seeing two very different sides of your ancestry, but only those two sides. All these people in the middle do not contribute to your Y chromosome or your mtDNA, even though they do contribute a lot to my DNA. So that's an important caveat to bear in mind. But if you're like me and you're studying the whole population, what happens is that you end up having other descendants of these other great-grandparents, and you start picking up all the lineages that exist in the population, even if you're only looking at one lineage for each individual. So you actually get back to looking at the whole population again. So this is a family tree. So those of you who are used to family trees might recognize it. It has the same structure, where this is time going down here. These are the descendants, these are the ancestors. Branches here, brothers, sisters, cousins, but they're not actual brothers, they're genetic brothers, genetic cousins. The time scale's much larger, it's not a couple of 100 years, we're talking about thousands of years here. And we're reading genetic variants, so-called markers on the Y chromosome. If you share them, you're in one group. If you don't share them, you're not in the same group. So you can use that simple logic to build a tree like this that relates different people. And this is a very special tree, actually, because a rather large group of very interesting people are related together in this tree. The chiefly lines of Clan Macpherson and of Clan MacGregor here are both related through this genetic family tree. And it looks like the chiefly lines of Clan MacKinnon, Clan Campbell, and Clan Macmillan also link in to the same tree. So you wouldn't expect this a priori. You would expect they all came from different ancestors. There's something special going on here. And it relates to the Northern Isles as well, because the Junsons from Orkney, that's not the Junsons from Shetland. There's two independent origins of this name. The Junsons from the East Mainland of Orkney link in here. The Tullachs, so that's the Tullachs from Shetland and from Orkney, because they are one genetic family, link in here. Moor is a name in both Orkney and Shetland. I've only managed to test thus far the Orkney Moors. They link in here. And there are a variety of families called Stevenson in Orkney that are different genetically. One of them links in here. So even up in the Northern Isles, we're seeing large families that link in to this tree. This is obviously not Norse. This is a Scottish or perhaps even Pictish side of our ancestry. I don't know where it comes from, but some people think that this might be the family tree of the royal house of Alpin, the so-called Shoal Alpin, or the seed of Alpin, because there are 7 families who claim this descent, and also MacRae, who are not included in the original 7. And out of these 8, 4 match into this tree. But I would also say that 4 don't match. So nothing's ever simple. But whoever this family descend from, he was obviously someone that contributed a great lot to Scottish genetics today, because fully 10% of all Scottish men fall into this family tree. It reaches 17% around Perth, Angus, and Fife, yet is below 1% in England. So it's something very interesting, and I'd like one day to try to work it out. So those are a little bit about Scottish lineages. What about the Norse lineages? This is a rather complex tree. I should have probably scrubbed off some of the different bits of it, but it's the same thing. We've got markers coming down here, a branch here, a big branch on this side, a big branch on this side, sub-branches, and you can see that there are a lot of different famous Shetlandic and Orcadian names here. Paulson, Georgeson, Nicholsons from Shetland, Sheens here, and then names from Orkney, Fletts all over the place, Bakey, Burger. What's interesting here is I tracked down people with the ancient and now extinct in Orkney surname of Papley, living in Saskatchewan in Canada, and another one who was living in Australia, and they both matched each other. Indeed, they match Nicholsons from Shetland. So this may well be the signature of Sigurd of Papley, who features in the Orkney Inga saga. Now, let's break this pedigree down and take the one side at the time so I can lead you through it. Now, my mother is a Flett, and this is actually a little bit of a pedigree of some of our family from the West Mainland of Orkney. I tested a number of Fletts in our family and Fletts in more distant branches, and we're very lucky that Storer Clouston has built some very deep pedigrees in this family. We descend from Alexander Flett here, and in fact I have Fletts from different parts of Harry, Fursa and Howen, who descend from brothers of Alexander, so we can infer that because they all descend from this Robert Flett of Bia, who died in 1681, and we have his will, that therefore this marker that unites the descendants, the 76-72 variant, is probably the marker of this Rob Flett of Bia, born about 1630. So that side seems to make good sense. Let's then have a look at the other side. We've got lots of Fletts here from Flotta, famous song, isn't it? Flett of Flotta, from Strumness, Orfer, Kirkwall, and so on. We even have Fletts from Banff. Some Fletts from Orkney went to the Murray coast. And even from Murray, they moved down to Lothian, where no one had heard of the name Flett, so they gave them the name Affleck, but they're genetically Fletts. So we think we can work this out, because I have some Fletts from Orfer, who descend from this fellow here, Jasper Flett of Hobbister. We have a deep pedigree, the Fletts of Hobbister in Orfer, in Orkney, and of Grudha in South Ronaldsay. So we're seeing that they all descend from this fellow here, Magnus Flett, who was a Burgess of Kirkwall, a rather important man flourishing around 1480. Indeed, his tomb, so we would infer, just as we did before, that the 6351 marker is probably Magnus Flet's marker, and his tomb is actually in the cathedral in Kirkwall to this day. You can just about read MF there. He has a crest here and his wife's crest here. It's hard to see, but it's 3 trefoils and a horn of plenty, very worn now, but it was used, I believe, to create this stained glass window in the cathedral where you can see the crest much more clearly. So this is Magnus Flet, the Burgess of Kirkwall. And we're thinking we're making sense of this tree, but there's a fly in the ointment here, because this Flet branch from Hunsketh in Harry also appears to descend from Magnus Flet. Yet, that's not what the family tree shows us. The Hunsketh Flets should descend from this Andrew Flet here, who descends from John Flet. And what Storer Clouston, the great historian, says, that John of Harry here and William of Hobbister, that's Magnus's son, were probably very near relations, because John appears on neither of the courts which tried cases to which William was party. Now, I know they're not brothers, but I suspect they might rather be uncle and nephew or first cousins. So this is not really making sense. It's not fitting together why this man is appearing on the wrong side of the tree. When we look to the tree, they only link together 4,000 years ago. So something is wrong. And what I think is wrong is that there's been an unken bull in the byre. One of these 3 women, Kristen Sinclair, Maggie Flet, or Kristen Hay, has slept with someone whose surname is not Flet. And little did she think that 400 years later, her very own descendant would catch her out. And I even know who she slept with. Because we've sampled essentially every Orcadian family, we can see it was a man called Taylor, or Tellier, as it's pronounced in Orkney, and they tend to live in Murwick in Bursey to this day. So we can learn many things from intensive study of families, but nevertheless, this family also descends from a Norse founder, just a different Norse founder to the other Flets. The deeper branches of this family are very, very prominent in Hordaland, so it would appear they come from Hordaland at some point in the past. So that's a bit of a run through the Viking genes. What about the non-Viking genes? Because we find a few of them as well. This is an interesting story. Some of the volunteers in Orkney belong to an unusual mtDNA group, a motherline group called L2C. And we found them in Flotta, we found them in Bursey in the West Mainland, Flotta in the South Isles, Firth in the West Mainland, and Orford as well. So scattered across a good bit of Orkney. But this is not a Norse type. It's not a Celtic type. Indeed, it's not even a European type. This is where this type is found. It's most common in the Gambia and Senegal in West Africa. And this was not the only, so I should say, I think these women did not have the same maiden names, and so they're not sisters, and because they're scattered across different parts of Orkney, it would appear to me it's been at least a couple, maybe 3 or more generations, since at least the late 1600s, that this African mitochondrial DNA has gotten into the Orkney gene pool. And it's not the only one, because I also found 2 families which had this U6 group that's only common in the Berbers of North Africa. So there's some very interesting but somewhat rare lineages to be found in Orkney. We haven't yet done the same analysis of Shetland, but I'll come back and report back to you when we do, if we find such things. So I was doing some research as to what this could mean, and we were looking in the Old Parish Register of Kirkwall. You can probably read it here. It's a baptismal register from 1750. It says, Kirkwall, April the 12th, a Negro usually called Neptune in the service of Mr. Fraser, officer in the military. So that's not the kind of a baptismal record you read regularly in the Northern Isles. And it seems that he had baptized into the Christian religion and taken the name Edward Kirkwall. And this was witnessed by the great and good of Kirkwall at the time. So it seems that there were slaves of African descent in Kirkwall, but Neptune cannot be the progenitor of these families because Neptune was a man, and these were motherline DNAs I found. So maybe somewhere a slave woman is actually the matriarch of a number of Orcadian families today. We probably will never know, but that's what it looks like to me. So people in Orkney are always daved by the idea that General Custer actually comes from Orkney, because Custer is an Orcadian surname. But I just want to point out he's not. He famously died at the Battle of the Little Bighorn in 1876, and people have traced his ancestry back to a place called Germantown in Pennsylvania, which kind of gives the game away. And indeed, a generation further back, you get back to Nordrhein-Westfalen in Germany. And various descendants of a great-great-great-granduncle have been tested as IL623. So I went to the Cursiter family in Orkney and had some of them tested, and they don't match him. So it proves genetically that General Custer is nothing to do with us. However, in doing this testing, I found something that I was not expecting and was very unusual. It's an extremely rare subgroup that the Cursters belong to. 5456 is only otherwise seen from these samples that are all from around Galway Bay in the west of Ireland, with typical Irish names, Corcoran, Conroy, and so on, apart from this one that is also Scandinavian. So I think it's speaking to us of some link at some point between Ireland directly with the Norse colonies in the North Atlantic. It requires more analysis, but there are a number of other families, particularly in Shetland here, the Inksters, but also others which show these specific Irish links. It's a small minority, but it's very clear that there is some Irish blood. So, I can't speak much about DNA these days without mentioning ancient DNA. I don't personally work with it, but I collaborate with people to allow them to compare the past samples to the modern samples that I have. In Shetland, there's only one skeleton that has given any ancient DNA, so there's not so much I can say with one skeleton from Unst. So you're going to have to listen to a couple of slides about Orkney. Hopefully we can get some Shetland skeletons and remedy that situation in the coming years. So Orkney people are very proud, as are people, I think, in Shetland, of their Neolithic monuments, tombs, stone circles, and so on. And I think they feel that they might be descended from these Neolithic people, but I'm not quite so sure. I've gathered together data from a number of papers such that we have 16 Neolithic men from Orkney. They're from all over the place: the Tomb of the Eagles, Westry, Sandy, Unston Tomb in Stennis, Rousey, and so on. And here's a very simple pie chart that just gathers together the numbers of people in different Y chromosome groups. And you'll see that they're not very diverse. A great lot of them all belong to this M423 or S185 group. And indeed, this little bit here, this segment of the pie, they weren't analysed in sufficient detail, but they quite likely also belong to this. So it's up to 3/4 of the population are all in this one group. And what I think's most amazing is to compare the picture when you look at male lineages to the picture when you look at female lineages. It's much more diverse. Look how many pie segments there are. There's way, way more. Almost every individual has their own lineage. So this means women have been moving around much more than men. There's some form of patrilocality where all the men, or not all, but the great majority of the men are related patrilineally to one another, even at different parts of Orkney and across 1,000 years of time, whereas the women are all different. It means women are moving more than men. Each generation a little bit more, and it adds up to this diversity. And this self-same pattern is seen in the Iron Age and in the Bronze Age. I'm not going to show you the pies, but it continues through time. So let's move forward now to the Bronze Age. We've only got 9 skeletons now, and you can see a bit of continuity. This blue type is continuing. Actually, the data are much poorer because a large proportion here, 45%, we did not get enough information to assign them properly. So they, again, could belong to this type. But we're also seeing a couple of new groups, this one here, and very notably this one here, the yellow group, M269. This is a very common group in Europe today, and it is thought to have come originally from the steppes of what's now Russia and was brought into Europe. Some people believe it was spread across Europe by the Beaker folk. Let's move forward another step into the Iron Age. The sample sizes are getting very small now, only 4. Some Irish Age men, some Pictish men from Bursey and from Westry. There's still one sample in the blue group, but now 3 samples, the majority, are in this yellow incoming group. And what's actually important to realize is when I look in my extensive data for Orcadians today, there's only one man of all the hundreds and the thousands indeed I've looked at who belongs to this blue group. So very, very few people in Orkney come down from the male lines of the Neolithic people who lived there. I am not aware of anyone in Shetland yet who belongs to this group. So there has been some sort of changeover in the genetics. It's not common anywhere, actually, this blue group. It's most common in Ireland, where it only reaches 1%. There's definitely been a big changeover in the DNA. And M269, on the other hand, is rising, and it's the predominant lineage in Orkney today. So now let's come forward one more step into the Viking Age. Sample size is only 3 here, but we have to work with what's published. They're very interesting. The picture's completely different. There's none of the blue group left. They're already gone. We may find more if we sample more. There's this green group here, which is a different Irish group again, showing that this connection to Ireland at least dates to the Viking Age. This is a very interesting one because it specifically links to Munster in southwest Ireland. And then we have these 2 groups here, the red one and the orange one, that are classic Norse Viking lineages that we start seeing arrive at this point in time. What does it look like today? Just for comparison, this is Orkney. So we've got this classic Viking lineage, another classic Viking lineage, got a very small number of Irish lineages here, and most of this rest here is Celtic in origin. Shetland looks very similar to Orkney. We have, this is a Norse lineage. These 2 are Norse lineages. Wherever S182 is hiding is a Norse lineage. This is also a Norse lineage. So there's a subset of Norse lineages, and the rest is probably rather Celtic in origin. So it's just to illustrate how DNA can change so dramatically over time. And it's actually the last 1,000 years, I think, where our gene pools have really become what they are today. And perhaps the things from all the way back in the Neolithic are less relevant to us. So we need more bones, and I'm hoping to start a new study with my colleague Martin Richards in Huddersfield, looking at lots of medieval skeletons to try to get into this area, and more Viking skeletons as well. I don't know if any of you know who this is. This might give it away. He's more famous in Orkney, just a little bit, than he is in Shetland. This is Magnus Erlendsson, St. Magnus. He's buried or interred in a pillar inside St. Magnus Cathedral in Kirkwall. There are in fact 2 bodies, one in one pillar who's a muscular, older man, thought to be St. Ronald, Magnus's nephew, and a younger, slightly built man with 2 severe wounds to the skull, thought to be St. Magnus. And my suggestion to the people of Orkney is that we should exhume these bones and do DNA and other analyses on them. We would learn so much. We could validate their identities. They should be uncle and nephew. We should be able to tell that from kinship analysis. We could do radiocarbon dating to help position them in the correct century. We could look at stable isotopes to learn about their diet and also their origins. Ronald grew up in Norway, of course, and I think it would be unique to know a medieval Norse nobleman's DNA. You know, is he related to the other Viking skeletons that are coming out of cemeteries in Dearness or in Rousey, or indeed any that we can find in Shetland, or indeed further afield in Iceland? Magnus's mother's father was Icelandic. We don't know who his mother's mother was, but she may also have been Icelandic. We can ask, does anyone in the Northern Isles today share this mother line with Thora, his mother, or is it found only in Iceland? Also, where is Magnus Erlendsson's Y chromosome found today? Are any of these families in Orkney or in Shetland carrying this lineage? We can also do fun things. We can use DNA to predict what colour his eyes were. Did he have brown eyes or blue eyes, red hair or blonde hair? I think these things have real scientific basis, but of course we need to bring the people of Orkney with us and show them that this is something valuable that we can do. It's not just about Magnus alone, because many of you will know that he is part of a much bigger genealogy of the Norse Jarls of Orkney, who ruled for about 300 years. So I think there's ample opportunity there for this Y chromosome to have slipped out into the population beyond the genealogy that we know about, and thus we may find it today. And what's more, Magnus Erlandsson, if you believe this very deep and borderline specious genealogy at the top, should have the same Y chromosome as William the Conqueror. The House of Normandy and the House of Orkney shares a paternal line. So we may find this Y chromosome in France or indeed in England, or we may not, because this genealogy might not be real. There are many, many questions that we could answer. As you will know, the Vikings spread all over the place, and I think it would be interesting from a named individual who we think we know who it is, to see if we could see where his DNA was found. Of course, there are many permissions that would need to be sought, but I wanted to put this idea out there to see what people thought of it. So, we spent the first part of my lecture, the majority indeed, looking to the past, using DNA. What does DNA tell us about our own personal futures? What is it going to tell us about health? I'm going to first go a little bit more into why we study Shetland in particular, and then take you through a few diverse examples of the sorts of things we can learn from cohort studies before ending up telling you a little bit about my new study. So, the main reason I think why people study Orkney and Shetland is because of a so-called bottleneck, and I'm going to explain to you what that means. We first have to think, how many people live in Shetland and Orkney today? 20 and something thousand in each place. Not that many people. But how many lived here in the past? It's actually, in Orkney at least, varied between about 10 and 20,000 for most of the last 1,000 years, with ups and with downs. And within that, we'd be interested in how many founders were there, how many people began this population, and how many immigrants have arrived in the meantime. And we don't know the answer. We don't have a Landnámabók like they have in Iceland, where they have the names of 3,000 mostly Norse settlers. But we do have a few clues. We have surnames. So in Orkney there are 260 Orcadian surnames, 100 of which are definitively Scots in origin. I mean, there's people called Cooper. I have no idea where the Cooper name originated. They made barrels. So some of them we don't know, but others we know that they're Scottish. So it's showing that we're talking about hundreds of founders, and I think there would be more than 1,000, but less than in Iceland. Of course, there will be founders of Iceland who are not recorded in the old books, but we're talking about somewhere in the low thousands as the number of founders, roughly. But in any case, it's definitely much lower than in cosmopolitan populations. How many people contributed to the population of London today? Thousands, millions. So what's actually happened is, upon the founding of these populations, they have gone through what we call a bottleneck. If you imagine this as the source population with thousands and thousands of people in Scotland, in Norway, and so on, and then only a small number come to the new place and found a new population. So it's like going through the neck of a bottle and expanding out again. And this bottlenecking is what reduces the number of variants that are found in the population. This is where they're lost. And this is what also changes the frequencies of the variants, because a small number of people can contribute enormously to the people who are here today. And these bottlenecks can be repeated. The Black Death rather famously hit Orkney and Shetland quite badly in 1349. There's evidence from land records in Orkney 200 years later there was still empty land that had not yet, the population had not recovered in size sufficient to farm all the land that had been farmed even 200 years after. There had been such a decimation of the population. So that's also a bottleneck that's going to change our DNA. So going now from prediction to actuality, I'm going to tell you about some work we did with the Viking study here in Shetland. We were able to get funding from the Scottish Genomes Partnership to read all the letters of the DNA code of 500 volunteers from Shetland. And we had samples from around Edinburgh, from Lothian, as an urban comparison. Now, I just want you for a minute to remember how much data we're talking about here. If I sequence myself and I sequence Brian, we would differ at 4 million different letters. So it's really huge amounts of data that are having to be crunched here. But most of these are seen elsewhere in the world in large surveys. People are sequencing Americans, Europeans, Japanese, all over the world. And we use a database called Nomad, which has 16,000 people in it as our sort of comparison to see whether a variant is common elsewhere or is actually rather rare. And what did we find? So first we had to chuck out those who were related. So we boiled this down to 269 people who were not closely related to one another. And we just basically counted. How many common variants did we find? How many rare variants did we find? And in fact, they look rather the same. Most typical average Shetlander had 3.5 million common variants, same in Edinburgh. Had 120,000 rare variants, pretty much the same in Edinburgh. But when we looked at the ultra-rare variants, so these are variants that were not seen at all in Nomad. So they're not seen anywhere else in the world, but they are seen in Scotland. And we can divide these up into singletons or doubletons. So singletons are variants that are not seen anywhere else in the world, and they're only seen in one person in Shetland, and they're not seen in Edinburgh. They are truly the rarest of all. And when we looked at them, Shetland had less of them. The typical person in Shetland had 2,000 versus the typical person in Lothian had 3,000. But when we looked at ultra-rare variants that were doubletons or tripletons or quadrupletons, so they're seen twice, 3 or 4 times, Shetland had 2.5 times more of them. So what we're seeing here is a large quantity of novel, unique variants in the Shetland population that are not seen in the rest of the world, and they tend to be shared amongst unrelated people in Shetland, not closely related at least. And it's speaking to this ancient shared heritage over the last 1,000 years. And it's not just the unique variants. When we look at variants that are seen elsewhere, they vary in frequency as well. Not the common ones or the rare ones, but the very rare ones, under 1%. Fully 10% of these were 10 times more common in Shetland than they were anywhere else. And of the ultra-rare ones, 2.1% of these were 10 times more common than in Edinburgh. Remember, the definition of ultra-rare is it's not been seen anywhere else in the world. So we can only compare to Edinburgh. So even compared to Edinburgh, it's not that far away, 2.1% were 10 times more common. What does that mean? It means 20,000 variants. And this is 400,000 variants. So it's not just 1 or 2. There are very large numbers of genetic variants present in Shetland that are either not present anywhere or are 10 times more common here. Overall, 1/10 of all the variants that we found are in this category. And this is quite good news in one way. It increases the power for me to find things. If you've increased the frequency of a variant by 10-fold, you've increased the power 10-fold to find out what that variant does. Most variants do nothing, but some of them are having profound effects on our risk of disease. But I think what's more important is if Shetland and Orkney is to benefit from the fruits of genomic medicine as genomics gets integrated into medicine, and thus that we can equitably participate in this future, we need to understand these variants and we need to study them because no one else is going to do it for us. Our genomes are slightly different from the genomes of other people, and we need to understand that and work out if that has any effect on our disease risk. It truly means there is a unique gene pool here in Shetland. And Smirk somehow knew this ahead of time because he made a cartoon, supposedly of me, saying, it's usually in the gene pool, you know. And I think I'd agree with him on this occasion. So you might ask me, okay, Jim, so we have all these different variants. What does it mean? And I don't know the answer yet, but we're going to spend time looking at it. What is true is that if there are all these different variants, some of them probably are medically relevant, and it would behove us to try to work out what they're doing. So I'm now going to take an example from the Viking study where we did something along these lines. During the recruitment process a few years ago, one of the volunteers shared a letter with us that she had received from the NHS, Clinical Genetics in NHS Grampian. And it said that she had a relative with a genetic disease called long QT syndrome that I'll tell you about in a minute. And it was inviting her to be screened for this disease. Now, it's important to realize how the NHS works. They do cascade screening. So if they have someone who has a disease, they will cascade out through the family to the parents, to the children, and to the siblings to check if they have the disease. And the ones that they find to have the disease, they cascade out from them, and they move through the family in this way, using cascade screening. It's a cost-efficient approach to try to find the people at the highest risk. Now, what is a long QT interval? This is an electrocardiogram here. So it's a graph through time of the electrical activity of the heart through a heartbeat and the next heartbeat. And I've coloured here in blue the QT interval. It's just a part of the heartbeat. And the disease is a prolongation of this interval. This is someone who's healthy, and here is someone who's got long QT syndrome. And you can see that this interval to the end of the T wave here is a little bit longer. It doesn't look like much, but this sort of disruption of the careful timing of the heartbeat can trigger very dangerous arrhythmias. So these can include fatal cardiac arrest, particularly in response to exercise or to stress or even just to a surprise. So there are various treatments for this ailment, including drugs or simply avoiding these triggers. If you know in your family that exercise can trigger this, you should avoid exercise. Leading up also to surgery, such as implanting pacemakers or defibrillators. As I said, this is a monogenic disease. One gene causes the disease, and in this case, the gene is called KCNH2, and it encodes a potassium channel, so a protein expressed in the heart which regulates the electrical signal that's controlling the heartbeat. This variant is exceptionally rare. In a very large sample of 100,000 people, there was less than one example. So it's exceedingly rare in the world at large. So we thought we would check our 500 people who'd had their whole genome sequenced, and we found one carrier. So this is not the lady who brought the letter to us. This is a separate individual. And we thought we'd better then check his relatives. So there were 42 of them in the study, but they had not had their whole genome sequenced. So we checked them, and we found that there were two others. This is the actual change here. Here you can see someone who does not have it, has a G here, and here someone has a G and an A in green. This is a carrier of this pathogenic variant. Here's the pedigree. This is the guy, and we can see he has a nephew who does not have the variant. He has a first cousin here who does. She has a daughter, and there are other family members, and there's more of them that I haven't shown here who do not have it. We can also infer that because their mothers were sisters, that they also carried this variant. So far, so good. But we then realized we could go a little bit further, and we could look at what are called haplotypes. So these are just strings of variants around a gene, and they're represented here by these barcodes, this digital representation. And we can see that so each person has two haplotypes, one from dad, one from mum, and so on and so on. And you can see, I hope, that these are identical. This haplotype, this string of variants, is the same as this one, because of course they both got it from their mothers, and the same as this one. And when I line them up, you see they're exactly the same. So we discovered that this was basically the signal for this long QT syndrome variant. We could then look in all 2,100 people to see if anyone else had this particular barcode. And lo and behold, we found two more people who carried this variant. But they were not related to the first family. We traced their pedigrees back to about 1770, whereupon it became rather complex. And so because they're not related, they could never have been found by cascade screening. They are not linked by other people into the first family. So either they're very, very distantly related, coming from a way back in 1700 or before, or there has been another Uncan Bull in the byre, and these people are actually related to the first family but don't know it. In any case, it's a really great example, I think, of the power of cohort studies, because we can scan a whole population without regard to who is related to whom and find these things that otherwise we would not have found. And it also gives the clinical geneticists a much more accurate idea of what the variant does. They are only seeing people who are ill and their close relatives. So they tend to overestimate the effect of a variant. When we screen the whole population, we sometimes find people who have the variant but are not ill. And it shows that not everybody who has the variant becomes ill. It's just increasing your risk of becoming ill. The clinical geneticists in Aberdeen told me that this result allowed them to give much more accurate counselling to the younger generation of carriers in Shetland today. And in fact, this variant is 150 times more common in Shetland than it is in other Europeans. It's truly part of the unique gene pool here. We are now extending this important survey to many other clinical genetic variants in Shetland and in Orkney, and I rather suspect we will find some more that are important. So, another thing we can do that pertains to our health is, rather than focusing in on the unique gene pool and individual risk, we can use genetics to learn about biology. Our cohorts are very richly annotated. We've measured all these different risk factors and measures that I told you. And if we compare these to the DNA, we can hopefully discover newly associated genes, new pathways that are underlying the disease process. I always tell people that genetics is a can opener. It's an incredible tool to get inside the black box, the things that we don't understand about the mechanisms of disease. We all know a little bit about mechanisms of disease. We all know that bad cholesterol increases your risk of a heart attack. But there are so many mechanisms that we don't understand, and it's only by understanding the mechanism that we can design new drugs to either stop what's gone wrong or to get what stopped happening to start again. So you need to know what's going on at the level of molecules and cells. So finding the genes is the first step on the road to new ways of diagnosing disease, because you need to know what's wrong with somebody accurately, and new ways of treating disease. And over the last 15 years, there's been a transformational approach in genetics called genome-wide association studies, or GWAS. And this was the first time that we as geneticists could look at all the genes in the genome in one experiment. So we didn't have to have hypotheses anymore. We could just look at everything. And it was just as well, because most of the hypotheses were wrong. So I'm going to explain to you very briefly and very simply how a genome-wide association study happens. So in this explanation, I have a cohort not of Shetlanders, but of giraffes. A small cohort of giraffes, and I'm not interested in blood pressure or cholesterol, I'm interested in height. So we measure the height of my cohort of giraffes, and then we get some genetics done, and we look at a genetic variant, in this case RS12345. So variants are just like cars. Cars have number plates. Variants have these RS numbers. They're just like a number plate to identify the variant. We go in and look, and this giraffe has a T and a C in the genetic code, a C and a C, and so on. And what do? The analysis is incredibly simple. We just order the giraffes by their genetic variants. And then we say to ourselves, well, do you think this variant has got anything to do with height? What do you think? I don't think so. It's completely random. We've got tall giraffes here, small giraffes here, tall giraffes, small giraffes. It's got nothing to do with height. So we test another genetic variant. RS123456 is its number plate, and this is an A to G, so we've got AAs and GGs and AGs. We take the giraffes and we order them, and we notice something different, don't we? It looks actually rather nice that all the AA giraffes are short, the GG giraffes are tall, and the AG giraffes are in the middle. This is a very strong signal that this variant is influencing height. And if we do this with humans and with traits that we're interested in, rather giraffes and height, this is how we do a genome-wide association study. But we don't just do 2 variants, we tend to do millions of variants. So the trait I'm going to talk about is back to our retinal vessels, the vessels in the back of our eye. And you can see here, I hope, that we've been measuring what we call tortuosity. So this is how twisty or wiggly the vessels are compared to the straight line distance. A bit like a river winds on its way to the sea, some vessels wind around like this. And it's considered to be a risk factor for heart disease and stroke, but it's not particularly well understood why. So we did a GWAS on human retinal vessel tortuosity. And this is the graph that comes out the end of it. So each dot is a variant in the genome, and there's loads down here. There are millions of these that I've plotted. And people call this a Manhattan plot because it reminds them, if it's worked, of the skyscrapers of Midtown Manhattan, as seen here. Up on this axis, we've got the evidence for there being an association. To what degree, when you line up the people in the order of their tortuosity, does it follow a nice line compared to the genetic variant or not? And along here, we've got all the genes in the genome. They're actually colored by different chromosomes, and these are all the different millions of variants. And I think you'll immediately see that the Empire State Building of this graph is here. And in fact, it's the only one that has sufficient evidence to cross this line of significance. And it's actually, these variants are in a gene called col4A2. So what is col4A2? It's actually a gene that encodes a subunit of collagen. So you'll all probably have heard of collagen. Collagen is one of the, I think, the most abundant protein in the human body, in skin and in so on. But this particular form of collagen is part of a thin sheet of fibers here under the inner lining of a vessel, including the vessels in your eye. And it actually has a very special function because it helps to control the passage of substances in and out of the blood vessel that affect how wide the vessel is. And this has important consequences for things like blood pressure, how relaxed or tight your blood vessels are. And in fact, it turns out that this self-same gene is associated with coronary artery disease. So it seems that we may have discovered a new role of COL4A2. And I wonder whether this might be related, that its role in tortuosity might be related to its role in heart disease, because windy vessels probably have more turbulent blood flow in them and may therefore have a more likely chance of a plaque, an atherosclerotic plaque forming. So there's lots to be worked out here, but I hope I've illustrated to you how we can learn about the mechanisms of disease using the data from the Viking health study. Now, the last GWAS I'm going to tell you about is about another trait. It's about lifespan, the ultimate trait. It's certainly the last trait that will ever be measured for any given person. And we've used the Viking study here and the Orkney study and many other such studies around the world. We did a little trick because happily it'll take a very long time for everyone in the Viking study to die, but we rather asked them how long their parents had lived. And they share DNA with their parents. And we were able, in this way, to put together data on 1 million parental lives and carry out a GWAS exactly the same as the giraffe height GWAS. Here we have the Manhattan plot. In this case, it's called a Miami plot because it's reflected in the waters of the Atlantic. Because we did 2 analyses. We did everyone together, and we did males and females separately, because we weren't quite sure whether men and women may die of different things or may have different effects on their lifespan. Women obviously live longer than men. And we were happy. We found 12 hits. So they've all been named here, the different genes. And they actually look to be mostly disease genes. Many people are chasing the elixir of life and some sort of aging genes. We have yet to find them. We did not see any aging genes in this analysis. But I think the most interesting thing that we were able to do with this was to build what's called a polygenic score. So polygenic just means many genes. So it's a score built up of many genes. And if you think, remember, we inherit half our genes from mum and half from dad. So at every variant, we have 2 copies. So we can, for each of these 12 variants here, simply count up the number of copies of the life-lengthening variant. So you've either got 0, 1, or 2. And then we just add these up, so you get a score that ranges between 0 and 24. If your score of 0, you've had the incredible bad luck to get none of these life-lengthening variants, and rather in the genetic lottery of conception, you've got all the life-shortening variants. Equally, if you have 24 as a score, you've had incredible luck in the lottery, and you've got all the life-lengthening variants and none of the life-shortening variants. As you will probably guess, most people are not as lucky or as unlucky, they're in the middle somewhere. And in fact, this is probably not that important for people in the middle. It's only important for people who are out in the extremes. And this methodology is now being used for cancer, for heart disease, and various such things. People say it's because genetics will soon be cheaper than measuring traditional things, traditional risk factors such as blood sugar and blood cholesterol. But what I think is much more important is that, of course, your DNA is fixed from birth. You could tell in children what disease they had a propensity to, and perhaps persuade them to alter their lifestyle, thus to lengthen their life and improve their health on that basis. So we did not study cancer or heart disease, but rather made a polygenic score for how long you will live. Here is the score. So here are different bins with the number of variants. So most people are in the middle. You've got a sort of normal curve here, and a few people out at the high extreme and the low extreme. And then here is the average lifespan of people in the different bins. So the score is working well. The people who've got lots of life-lengthening alleles are living longer. The people with less life-lengthening alleles are living shorter. But what was really incredible to me, actually, is when we took our score into a brand new population, Scottish individuals, and we calculated these so-called survival curves, Kaplan-Meier plots, we saw an amazing thing. So I'll just take you through it. The red and orange lines are for females, and the blue lines are for males. And you can see that females live longer than males. The line is further this way. So everyone is alive at age 40 in this study, and pretty much everyone has died by age 100. But what we've plotted in the two lines per sex are the top and bottom tenths of the polygenic score. So the top extreme and the bottom extreme of each score. And you can see at the average probability here, the women with the high end of the score are living 5.5 years more than the women at the low end of the score, and it's exactly the same for men. So not taking into account smoking, diet, exercise, employment, we can explain 5.5 years of life with your DNA alone. Isn't that incredible? Think of the enormous contribution of chance to when you die. You trip up and break your leg when you're 90. It's usually the end of you. But still, despite all that, with this very large data set, we can see these underlying propensities. And it's not to say that it's not to do with smoking. Of course, your genes influence how much you like smoking, how much you like taking exercise, and what you like to eat. So it's actually not nature versus nurture, it's nature and nurture all mixed up together. So I don't know where this will get us, but maybe one day when I retire, I'll be able to take a test and work out how long I've got left and how much I should spend on my pension or how much I should be careful and save it. We don't know, but there are amazingly interesting things, I hope you'll agree, that we can do with DNA. I've tried to illustrate a whole variety of these today for you with these platform resources that we've built up in Orkney and Shetland, both in dissecting diseases and in dissecting our history and learning more about where we came from and when. So what do I want to do next? Well, I've been funded by the Medical Research Council to quite a grand tune to recruit 4,000 new volunteers who have ancestry from Orkney and Shetland, specifically people with 2 or more grandparents from either Orkney or Shetland. One from Orkney, one from Shetland's totally fine. They don't have to live here. They can live anywhere in Aberdeenshire, Edinburgh, London, Canada, New Zealand, wherever they may be. And this is a much lighter touch study. Those of you who attended the Viking study, thank you very much for coming and giving me lots of your blood, fasting all night beforehand, coming again and being measured for 2 hours, and filling in a questionnaire. This is much simpler. You hear about the study, you volunteer, you get sent a code in your email that takes you into your own private part of our database, where you then read about the study, consent to the study, and start filling in the questionnaire. You can take a break from the questionnaire, come back to it in the morning, whatever you want to do. It's all done online, on your iPad, on your phone, on your computer, in the comfort of your own home. When that's done, we send out a little saliva kit, a spit kit. You spit in that, post it back, and you're finished. Very, very easy. The reason I want to do this, of course, is to expand the numbers, to increase the power. I keep telling you I'm interested in these rare variants, particularly these variants that are found much more commonly in Shetland. But because they're rare, they're hard to study. If I recruit 2,000 folk from Shetland, I've probably only got 10 or 20 people with this variant. So if I double the numbers overall, I double the numbers with the variant. I might get up to 40 people. And it's so much easier to see what's going on when you've got 40 people than when you've got 17, believe me. But there's another thing that we're doing different here, because we're asking people for consent so that we return to them the actionable findings that we find through our collaboration with NHS Grampian. So what is an actionable genetic finding? It's basically a gene change, so a variant that is linked to a serious disease, but for which there's something that can be done. There is a preventative action that will allow us to either delay or improve the outcome through NHS treatment. And in most of these cases, the earlier you know about it, the better. It allows us to enhance the preventative measure. So it's definitely better to know about these things. We estimate that very few people will have this. In other surveys, about 2% of volunteers have had an actionable finding. And these might be things like a high cholesterol gene that runs in your family and greatly increases your risk of having a heart attack, or long QT syndrome. This is the disease, you know, when a young man of 25 is running about playing rugby and he drops down dead, this disease. Or perhaps a breast cancer gene such as many people hear about these days. These very important things that are very important to know about. So what would happen in that case is that we'd pass our volunteer on to NHS clinical genetics that look after Shetland, and you'd follow up the normal pathway of care, which would actually involve a repeat DNA sample to be totally sure that this is actually something we're seeing in your DNA. It's entirely optional. You can join the study without saying yes to this bit, but the great majority of people so far have said yes. It's important to realize also that we're not analyzing your DNA in a one bite. It's very expensive. So we wait till we've got 2,000 people and then do a batch. So it may be some years before we know anything about anybody's DNA, and at that time we would speak to the clinical geneticists in Aberdeen. And I want to say to anyone in the audience who is a Viking 1 volunteer and would like to avail of this, we have a lot of red tape, but we are committed to offering the same opportunity to people who have been so kind as to join our earlier studies. But bear with us, it takes quite a while to get permissions and to clear the various governance hurdles. We are the only study in the UK that has this permission so far, but I think it's a great opportunity, particularly in Shetland, where we have such interesting variants. Just to close, I'm going to show you two contrasting stories of what people have done when they're faced with this. The first comes from Iceland, who are ahead of us, as always, in many ways. Iceland has a large company called Decode Genetics. They have 100,000 samples now. And as such, when they were doing analysis, they also found a cancer gene and a cancer variant, to be specific. And they now know of over 1,000 women in Iceland who have this variant and who I think ought to know about it. However, the Icelandic regulators determined that without explicit consent, because these women had not consented to this, because they didn't even know that this cancer gene existed, neither the government nor private industry can notify people of these risks. Now, the CEO of Decode is a man called Kári Stefánsson, and I tell you, you wouldn't need a DNA test to know that he is descended from Vikings. He is a towering presence in many ways, and he said, that is utter, thorough bullshit. He said there's a tradition to save people in life-threatening conditions without asking them for informed consent. Why should it be any different if that risk comes from a mutated gene? And I must say, I do believe this as well. He solved this puzzle by setting up a website where people could opt in if they wanted to know their BRCA2 status, their cancer gene status. So it's sort of sidelined this. Coming to the UK now, there's a wonderful, enormous study called UK Biobank of 500,000 people, and they are also now reading through the DNA and will find many people with actionable findings, but they have no permission and no plans to return these findings. So I think it puts people in an invidious situation to know something about someone and not be able to tell them. So I'm very happy that we got permission to feed this back to the populations in Orkney and Shetland. So we are not in this position because people have consented up front to have this information returned if they so desire. So if you're already in Viking or indeed Orchids, there's no need to join. We already have your data and we're very thankful for it. So if you got a leaflet and you're in, please hand it to someone. I'm sure you have sisters, brothers, offspring, aunties, parents who are not in the study and might need a helping hand, particularly men who are not so good at coming forward. By doubling the number of volunteers, I've already explained to you, it'll help us so much to understand these variants that we're finding. Each variant is rare, so we need many people to have enough of each one. We will use the electronic health records to see what these variants do to people, how it affects people through time going forward in the next 5 and hopefully 10 years. And I hope that you'll be ambassadors for me and try to encourage as many people as you know to join up. It will help medical research, and it's really a chance for them to find out about their actionable findings as well. If you want to hear more about our work, we have a snazzy new website here at edacuk/Viking. That's updated from time to time with when new papers come out and new discoveries. Those of you who follow social media, please follow us on Facebook, Twitter, and so on, using these tags. All it remains then for me to do is to thank the enormous number of people in Edinburgh and all over the place who've been involved in both the collection of these data, the analysis of these data, the writing up of these data, the many funding bodies that have contributed through time to this, and of course, most of all, the volunteers for the Orcades and the Viking studies, without whom we could never learn any of this information. Thank you very much.